Hey guys, welcome to another episode of the You've Got Potential podcast, where we talk about graduate school and the undergraduate experience in research.
Today, we also have our guest, Justine, here with us.
Hi, everyone.
I invited Justine to this podcast because she was the first person I knew who was part of undergraduate research. She was my not roommate, suite mate in freshman year. And I feel like she already was in a lab when we got there. It's not a lie, right? You were pretty much there already. And so she was like ahead of all the curves and definitely helped me in my journey to like get started in research. So she was an inspiration to me. So she had to be on this podcast. And so Justine, would you like to introduce yourself and a little bit about what you do?
Yes, thank you, Monica. That's very sweet of you. So I am a junior or I guess a rising senior now, and my major is molecular biology and biochemistry, but I have delved into the humanities a little bit with a minor in history. And would you like me to explain a little bit, just like a brief overview of my research at this point? Okay. All right, so I'm working with Dr. Yu at the Center for Alcohol Studies. But funny enough, my research isn't really alcohol related at all. It's a kind of unique project. So we're looking at the source of neuroinflammation in painful diabetic neuropathy. So I'll touch on blood cells, white blood cells a little bit, which are essentially, I'd say, the bodyguards of the immune system. So they're able to travel through the blood and tissue, and they really try to keep out foreign invaders. So our research centers on the role of chemokine, CC motif chemokine receptor 2, which really heavily influences the movement of these white blood cells. And we think that there are changing, not changing, but rather variable levels of CCR2's isoforms in different individuals. So that might actually be influencing how well drugs might, painful diabetic neuropathy drugs might be able to target CCR2 and really play a big role in its development and the way that we're able to, effectively treat that condition.
Thank you. Could you just give like a quick definition of what painful diabetic neuropathy is?
Yes, so there are a couple different types. We're mainly focused on peripheral diabetic neuropathy. So it basically travels, it starts in your extremities first. So it's called distal to proximal. So the severity basically increases. It'll start in your hands and your feet first, and then work its way up to all the major nerves and axons there. And then a lot of people tend to feel either a numbness or pain in relation to things that wouldn't normally cause you pain. Someone might touch you with a pen, and that might cause you to feel a sharp pain. So there's a little bit of a disparity there. And then some people also develop ulcers just because you're kind of less aware of your footing. It's easier for you to kind of step on things, which is something I didn't really consider as one of the major symptoms, but it is out there.
Do you want to go over, because I've always been curious about this, sort of the origin of nerve damage, and what would it be, type 2 diabetes? From what I understood, right, it's like you'll have excess glucose in the blood, right, and that'll be hard on the arterial walls, and that might damage the blood supply to these nerves. Maybe you have a more in-depth perspective on how that happens and the mechanism of that.
Right, that's exactly it, Venkat. You summed that up pretty nicely. I would say that a lot of this isn't really too well known, I would say. But we do know that inflammation is one of the major factors now. So that's definitely coming into play in a lot of these diseases, especially like autoimmune disorders. It seems like really that the case is that basically the inflammation is causing damage to neurons, and then this is really one of the main causes of neuropathic pain, but the mechanism you explained seems to be the primary one.
Okay, okay, cool. So what made you interested in this research or want to join this lab specifically? What maybe walk through some of the interests you had going in. It seems like you joined the lab early, so walk through some of the interests you had coming into college and how that aligned with your choice.
Right. I would say my lab found me a little bit more than I found the lab. As Monica mentioned, I got in pretty early, but I did have a friend who was in the building, and he mentioned that Dr. Yu was looking for a couple freshmen to start. So I came in, and I thought that the premise was a pretty interesting lead to really study inflammation and neuropathic pain, which overlaps pretty nicely under the umbrella of painful diabetic neuropathy. And then I also thought it was interesting that it's really this idea, there's so many inner mechanisms and pathways in the body that, I'd say like 90% of the time seem pretty aligned and in sync. But then there's also this idea that it's not infallible, because inflammation itself is really the immune system, it's trying to protect itself, which it does fairly well. But sometimes it perceives these risks that aren't really a risk, or might overreact to something, which is the case for a lot of autoimmune disorders. So that was pretty interesting to me, just these little slip ups and how one small thing can really cause, this monumental damage.
So I want to ask then, starting your project so early on, right, as a freshman, um, what was like the most challenging aspect, considering that you probably didn't have too much coursework under your belt? Maybe you had some introductory biology, but probably not much more than that, right?
Yeah, you're absolutely right. I think the hardest part is maybe I didn't realize there was so much troubleshooting involved. Because I guess I kind of figured that protocols were pre-made, which is completely not true. Even if your kit comes with a protocol, that's probably not really what you're going to be working with. So it did take me a while, but it was lucky that the group I was working with, this project was pretty much in its infancy. So I've been able to see pretty much from scratch how we came up with reverse transcription protocols, RNA extraction. And that was a pretty tough process just because we're working with blood. So when I first saw that, I was like, man, why is it so chunky looking? So there has been that. I think just the long process of really perfecting your protocol until so you're able to get results, where up until then, a lot of things might have gotten in the way.
I was going to ask, what's something that you did as a freshman and did it so either it took so long, or you did it so or you did it so wrong that is now just second nature, or just something that becomes so easy to you and makes so much more sense?
Yeah, something very dumb that I did. I would load, for some reason, I didn't think it was necessary to add loading dye to, anything I did in the beginning. So then I had no idea where anything was, and it would just run right off the gel, which seems like the first thing now. Oh, I can't really see anything that I pipetted here into the wells. So I guess that would be my biggest blunder.
You mentioned in terms of finding your interests and how you're applying it to your path at Rutgers, how you basically are pioneering your own way and combining different things, like your MBB major and your history minor. That's something that we don't hear about often. And so how else in the future, per se, are you taking interest or taking motivation from? What are your career goals and how are they implemented into your research specifically?
Okay, So I'll just touch briefly on the fact that I'm very lucky to have an internship this summer with BMS. Yeah, thank you. So that has been, this is in the discovery toxicology group. So that's really something that I had no expectation or knowledge of really what this sort of science did. I think a lot of people have this misconception that it's somehow involved with forensic science, or, I guess I watched too many CSI shows at this point. But it's really kind of interesting to see how this part of the pharmaceutical industry is not only looking at drug candidates to see what's feasible, but also in what levels are they safe and effective. So it's really bridging the gap between drug discovery and the non-clinical safety group. So that's definitely something that is a whole new branch of science, really, for me. Some of their methods are pretty cool. I've learned a lot of cell culturing and different assays. So a lot of things along that line. So that's definitely something I'll be bringing in to inform a lot of my future career decisions, I think. Because my ultimate goal would be to be involved with pharmaceutical research. So just to get a sense of all of these different groups really has been helpful. Before, I would just kind of name, yeah, let's do pharma, not really understanding how many really steps there are to the whole discovery process, and you can be involved in a lot of places along this drug discovery pipeline.
Thank you. So that's how like your internship is like helping you move forward in your career path. But has your undergrad research at like your lab with Dr. Yu, has that played a role in also shaping your interest to like go into pharma?
Yeah, that definitely has. So I'll bring in something else here, which is we have been looking really at one particular CCR2 antagonist, which has been developed by AstraZeneca. So this drug has been shown to interact with CCR2 effectively, but there's also been kind of an element where it didn't reduce pain any more than the placebo. So that's something that we were interested in, because it seems like there's a connection here. There is definitely interaction happening between this drug and the molecule of interest, but we kind of were a little bit baffled by that. So one of the things that we're considering is the fact that maybe there might be some interplay between basically the levels of CCR2's isoforms. So there's an A and a B that have been previously published about, but then there's a third isoform, C, which our lab has just started looking into. So there's a couple things going on here where we're really trying to quantify the levels in a small group of patient blood samples and try to understand if there's any ratio differences, which there has been shown to be quite a bit of variability between just A and B. So this semester or the next, hopefully we'll be able to see how C really fits into this mix. And I think this, to answer your question, Monica, I've delved off a little bit back to the science, but I think it's definitely interesting, because I would consider this more like pharmacogenetic work, really trying to understand how the individuality of patients and just their genetic makeup can contribute to the effectiveness of drugs. And I think that's kind of looking a little bit towards precision medicine, which is something pretty cool that's really up and coming in the field.
Cool. So when it comes to your lab specifically, how are you studying different forms of CCR2?
Right. So we're really trying to understand the expression levels using reverse transcription, PCR, and then of course you get your gel. So then from there, we've been able to take basically photos of the gel, and we use ImageJ software, which is pretty handy from the NIH. So that's been one of our best tools to really, we generate a peak from whatever it is was inside, right? So we, at this point, we've also done some sequencing, so we know that whatever it is that we've isolated from PCR is that particular gene we're interested in. And then we're able to look at the size of the peak and attribute a number to that. So we're able to kind of create a ratio for each patient sample. And in that way, we can understand how these levels are differing among individuals, both with painful diabetic neuropathy and also diabetes, but no pain. And of course, our control, which has neither.
Maybe you could do some sort of explanation as to how these different isoforms of CCR2 or CCR2 in general, how that affects the treatment or the studying of painful diabetic neuropathy.
Yeah. So this particular, or I'll go back a little bit further. So basically, we do get isoforms from alternative splicing. So there's a possibility that you know, when the DNA is actually formed, you know, you have cuts in certain areas. So maybe introns that are taken out and removed in some instances aren't actually removed in others. So you can get basically unique sections of an exon now. And then we found that that's the case in CCR2B, where it actually has, I don't know the base pair length exactly, but it seems to be a significant chunk of unique DNA that's not found in CCR2A. So whenever you have, you know, a sequence that's now totally unique or just additional, then we're looking at something different. We don't really understand how this would interact. Maybe a drug wouldn't be able to interact the same way with one isoform versus another variant. And now we know a little bit more about C, where it's actually very truncated. So it's possible that it may have been able to bind to the CCR2 antagonist, but then you might not have gotten any reaction because it seems that it's been kind of cut short, so it may not be functional.
What is the significance of CCR2 to treating painful diabetic neuropathy?
Yeah, so really there's, I guess the key link here would be that CCR2, because it's able to perpetuate inflammation, and it's a really strong mover of really monocytes within the body, there's a lot of evidence that CCR2 may also play a role in painful diabetic neuropathy. Right, so CC motif chemokine receptor 2 is able to recruit white blood cells, and they're bringing these to inflammatory sites. So really, chemokine receptor 2 has a lot of influence over their mobilization. So this has also linked CCR2 to a number of central nervous system diseases. And then we also understand now that it has been a perpetuator of neuropathic pain. So we're able to see that in a number of studies, when the CCR2 receptor gene has been knocked out, neuropathic pain has been really significantly attenuated in a lot of preclinical models. So this is something that really overlaps in painful diabetic neuropathy, because we understand that inflammation is causing the development of neuropathic pain in individuals with painful diabetic neuropathy. So CCR2 as really a source of this inflammation and thus the neuropathic pain, it has been our starting point. We think that there's a lot of evidence to suggest that it plays a major role in the development of painful diabetic neuropathy.
Just out of curiosity, have you read any papers or maybe seen any work from like students or professors of the structure of the CCR2 protein, or maybe some of its ligands?
So there has been a lot of research that does center on the fact that functional differences between A and B have have led to differences in their actual signaling. So that is known, but it's not as clear. Really, there isn't as much research that focuses on really their structural aspects. There's only a handful that touch on A and B, to my knowledge.
Okay, yeah, I was just wondering. My work this summer involves like managing a database that keeps like all like these protein structures reported by different scientists all over the world. So just as a shameless plug, check out the RCSB database if you're interested in the CCR2 protein structure. I'm sure they have it. I'll look it up.
I am going to follow up on that plug. You are a undergrad reviewer, right? You're still okay. You're an undergrad reviewer for the journal. So if you would like to just either as an author explain how that process went, or as a reviewer, if you'd like to give people an understanding of what that's like, for a shameless plug.
That would be great. I don't mind at all plugging the journal. Yeah, it's been a pretty interesting experience to see it from both sides, both as an author and actually know what the authors are going to receive from the undergrad reviewers. So it is I think a 13-week course. So they really teach you quite a lot of the principles of to have a basic understanding of research ethics. How should the logic and the structure of a paper come across? So the flow is really there, and it does mimic, some of the high caliber papers that you guys have probably been reading. So though much shorter, right? So we've been able to really bring together a big team. This has only been the second year that the journal has been at Rutgers. So there's 3 steps really to the process, where first your paper is coming in all its raw glory, really, to a group of reviewers. So we're split up into different sections, and there will be like 4 or 5 people that get one paper at a time. So you're able to reflect on it a little bit, come up with maybe structural issues that you have. It's possible that you think one section might flow a little bit better if it was moved to a different part, or maybe some sentences aren't as clear. And we really want to bring it down to a level where everyone, no matter the discipline, is able to clearly understand what this is, whether it be, a music oriented paper that's focusing on Bach and his feud with someone else. I don't know anyone but Bach at this point, but yeah, so something like that, or you could have a really heavy science oriented paper that's touching on mouse models, and we want everyone to be able to relate and understand these basic principles that are inherent in each of these fields. So that's definitely been something to be really proud of. I think, to be involved in the publication of the journal. And yeah, on the author side, now I'm dealing with how particular I know my group of other reviewers is, and I'm getting all of that feedback. So it's definitely been helpful in improving my writing skills, and it's enhanced me in both places. I think my ability to really look critically at a paper and also benefit from that feedback.
Was it a stressful part of your coursework or anything?
No, I wouldn't say that. It's actually been pretty enjoyable to kind of have this little group that meets. It's fairly informal. So I would say not a big contributor, but you do want to make sure that you've had time to read the paper and reflect and think about things that wouldn't really involve any rewriting, just small points where the author could enhance aspects and make it clearer for the audience. So yeah, I would encourage anyone to join. I think it's a great experience, and I'm so glad you're part of it now too, Monica.
Yeah, I'm actually pretty happy with the way they're structuring it. Like for people who are in the reviewer program or as a copy editor, they're very good at they're very conscious of like our time and like the fact that we're also students. And so the way they schedule things is very like spread out. It's not too, at least in my experience, it's not too like in your face, like we need it now kind of thing. As long as you're okay with managing your own time. And yeah, it is a fairly new journal. So I think they're managing it very well. So if you want to be a part of you want to be an undergrad reviewer, I will link the application to next year's batch. It's due on July 30th, I believe.
I think, maybe we want to go, like, if you don't mind me asking, Justine, like the future plans. Like I assume you want to go to graduate school, conduct research. How's sort of like the process been going so far? What have you been looking forward to?
Yeah, I have been looking at grad schools. I think the major question now is just master's versus PhD at this point. I think
This is a great topic.
yeah.
This is a good topic. Yeah.
Yeah. Because it seems like for a PhD now, as I'm really reading over these other personal statements and statements of purpose, whichever they're called, yeah, I've been really going to town online and seeing all of these really specific statements of purpose. So I'm not really at the point where I can pinpoint exactly, okay, this is the model system that I'm set on, and I know that I'm interested in this disease. I think my interests are a little bit broader at this point. So it seems like a master's fills that gap a little bit better. Most tend to be, I'd say, no longer than a year. That seems to be the average. So about 30 credits spread over 2 or 3 semesters. It might be a little bit more than that at some universities, but so all of these classes are really, I think they do fill that gap, and they're able to show a different level of coursework where I think I'd be able to get my feet wet there, kind of pinpoint more of a clear idea of a PhD project. And that's something that's pretty long term, right? Most people tend to stay in a PhD program upwards of 5 years, 6
Right.
if you're really lucky. So that seems like a pretty definite step to take, where you're very set of your PI and where you're going to be in terms of the university.
But I'm glad that like you considered the master's. One thing I wanted to ask was, and for people who are also considering a master's, is if you finish your master's and then you either stay in the same like school or you decide to do your PhD like elsewhere, do you still have to take that like first year or two of coursework? Or how does that like, does anyone know how that works?
Oh yeah,
Because it would be, yeah.
I have somewhat of an idea. So I think I researched this very question in the past, and I can't speak for all universities on this front, obviously. But from what I understand, generally there are some courses that are PhD specific that you might have to take when transferring from a master's to a PhD, but there tends to be a very strong overlap in that 1 to 2 year period that's focused mostly on traditional coursework, whereas the PhD stuff afterwards tends to be more focused on lab work.
Yeah, I only ask because like one good point when you're considering a master's versus a PhD is that like Justine, like once you go through your master's, if you want to do your PhD and you're settled more or less on a project, it would be really annoying if you had to do like an extra 6 years. People, this is a big thing too. I'm glad you said 5 or 6 years if you're lucky, because a lot of people think it's 4 years, but it's not. It's not. They try to trick you like that. It's 5 or 6, at least in STEM. Yeah. Thank you guys so much for watching. Justine, like thanks so much for coming on. Like you were so positive, so informative, very clear. This is always like a fun time. Yeah. This is always a fun time. So thank you guys again for watching, and just remember that you've got potential.